Okay, it is my great pleasure to introduce Dr. Pennings, Assistant Professor of Biology at San Francisco State University, and hopefully not for long, Associate Professor coming up, right?
No, I'm associate. I'm going for full.
Oh, did I misread your? Okay, Associate, sorry.
It's
Associate
okay.
Professor going for full, and that makes a lot more sense given this awesome record. Dr. Pennings completely skipped her bachelor's degree and went straight to the Master's of Science at the University of, oh no, that's not quite true, but I guess they don't have a bachelor's degree at the University of
Sure.
Michigan. You go right to the master's degree. And then did a PhD in evolutionary biology at Ludwig Maximilians University in Munich. Also had several postdoctoral positions in Munich, in Harvard, doing population genetics of HIV, and at Stanford, modeling the evolution of HIV drug resistance. Dr. Pennings is highly honored with the number of, especially I was impressed to see the number of different films and videos that won awards over the time, including some of my favorite titles, Why Do Slave Ants Kill Slave Masters? And Good Milk, Bad Milk, which I thought was great. But also, Dr. Pennings won the Omen Prize, given by the International Society for Evolutionary Medicine and Public Health for, and I quote, the best article published in 2016 in any scientific journal on a topic related to evolution in the context of medicine and public health. And I would just retire after that. It's so amazing. What a fantastic publication record. I encourage you to check out Dr. Penning's publication, but many articles in PLOS computational biology, which I never get into, so that's exciting. PLOS genetics, PLOS pathogens, doing a whole PLOS library apparently. Doing G3, E-Life, and evolution, among a bunch of others, and really impressively has gotten about more than $5 million in funding for all the research that she's doing at San Francisco State and all the great students that you're mentoring. So there's a lot more I could say, but I'd rather hear about what Dr. Pennings has to say. So let's go.
Thank you. Thank you so much. So hi, everyone. I'm really excited to be talking here. This series of seminars has been really fun to learn about what different people in computational biology and bioinformatics do in the CSU system. And so I'm excited to share some of my work. And I want to share my screen, and it's going to take a minute because my iPad is slow and it needs to connect to my computer. But when I have my iPad screen shared, I can show you my slides and I can draw on the slides, which I like. It's like, it's as if I have a little whiteboard.
We can see it just fine.
Okay, that is cool. Okay, so I am going to be talking, I think I changed the title, but the topic is basically the same. I'm going to talk about a model, and the model will help explain coexistence of resistant and susceptible pathogens. So I'm interested in drug resistance. And one of the big questions in the world of drug resistance is, why do we have, for any kind of pathogen, why do we have both resistant and susceptible strains both existing at the same time? And I'll explain why that is a question, and I'll explain how I think, how we can explain it. Okay, so, sort of very brief introduction to the kind of work that I do. So I'm interested in data, I'm interested in models, I'm interested in drug resistance and evolution and population genetics. Population genetics is when we think about how genetics plays out at a population level. So, for example, one of the big questions in population genetics is, if one individual in a population, if a mutation happens and that mutation, makes that individual more fit, how does that mutation spread through a population? Now, in the case of drug resistance evolution, this population can mean different things. It could mean what happens, within a person right? So if I'm infected with a virus, there's like a whole lot of virus inside of me, and that virus can be considered a population, right? And so evolution can happen inside my body. The virus inside my body can evolve. But the population, in other situations, we have to consider, a whole bunch of people that may be all infected with something, a population. Okay, and so we'll, in my work and in this talk, sometimes we'll go back and forth between population as in within a host and population as a whole bunch of hosts. And so sometimes that makes it a little complicated to keep track of what level we're talking about. Okay, so drug resistance. Let me very briefly tell you what drug resistance is in case you are not familiar. So this image is from Wikipedia. And so here in blue is like what we hope would happen. We have, bacteria, or it could be viruses or any other pathogens, something that we don't like. They multiply, right? You're infected with something, it's multiplying, it's making you sick. We use drugs, you get treated with antibiotics, and hopefully the bacteria die, or they cannot replicate, and your immune system will have a chance to clean them up. That's the sort of the normal thing, the thing that we would like. But unfortunately, sometimes these bacteria, their genome changes a little bit. Again, they multiply, but now if we treat with a drug, these bacteria are resistant, the drug doesn't do what it's supposed to do, and the bacteria can keep on growing. Okay, so drug resistance basically means normally the drug would work, but now the drug doesn't work anymore. It doesn't kill the bacteria, or it doesn't stop the virus from replicating. And obviously that's a problem because we depend on antibiotics and other drugs to keep ourselves healthy, right? And that's especially true for situations where people are already vulnerable. For example, if you are being treated for things like cancer, or if you have like immune problems, or if you need to get a transplant, we need antibiotics to keep those, to keep the pathogens under control. And so if the pathogens become resistant to the drugs, that is a real problem. So drug resistance is a problem in viruses like HIV, bacteria like Mycobacterium tuberculosis that causes tuberculosis, Staph aureus. If it's resistant to penicillin, it's known as MRSA. E. coli, that's what I'll be talking about mostly. Fungi like Candida, there's a lot of resistance that's problematic. And then parasites like Plasmodium, the malaria parasites, there's a lot of resistance there too. So it's something that occurs like in many different types of pathogens. Okay, so a lot of my work over the last decade or so has been about what happens inside a person within host evolution. And so this image is from a paper that was largely written by Alison Feeder, the first author here. She is currently an assistant professor at University of Washington in Seattle. This is actually data from another study. Like I'm mostly in the business of reanalyzing data that other people have collected, and that's what we did here as well, or that's what Alison did here as well. And what you see here is every line here is data for a single person that is infected with HIV. And what we're measuring is how much of the virus inside the body of that person is resistant to a certain drug. All of these people are treated with the same drug. They're only treated with one drug. This is old data from the early 1990s, before we had like the amazing combination treatments that are now common for HIV. Okay, so here is the number of weeks on treatment, and here is the percentage of the virus in those people that is resistant. And so what you see is that, you know, they all start at very low resistance, like the virus is not resistant to the drugs, but then very quickly, within a couple of weeks, you know, all of those lines go up, showing that these populations within these people evolve very quickly to become resistant to the drug that they're treated with. And obviously, this is really bad news. It means that this drug really doesn't work, because within a couple of weeks, the virus has become resistant to the drug. And, you know, obviously that the drug then doesn't work anymore. And so the treatment has become useless. This was, you know, very common in HIV for maybe the first decade, that we had drugs for HIV, but the virus would become resistant so quickly that basically the drugs were useless. In the mid-1990s, people started treating HIV with multiple drugs at the same time, and it worked much better. And it worked much better because resistance evolution became much less common. Okay, so that's what happens within a host. But then if resistance evolves within a host, that resistance doesn't necessarily stay in that person. Now there are resistant viruses, you know, all over that person's body. And so if that person infects another person, now the resistant strain may be present in another person, even if that other person has never been treated with that drug. Okay, so that's what we call transmission of resistance. Transmission of resistance is a problem in all of those different pathogens. And this is just one example of people who've been, who study MRSA strains. So MRSA is methicillin-resistant Staph aureus. And these strains, you know, are known to sort of travel. You know, they infect one person, the person takes an airplane, and the strains go all over the world. And so these are just some of the, you know, these arrows indicate some of the known sort of migration paths that these resistant strains have taken. Okay, so the problem of resistance is sort of twofold. One is that resistance evolves within a patient, and the second is that it spreads between patients. And if it spreads between patients, it can go all over the world. Let's see, this is a little bit more about MRSA. So people have been, have also tried to sort of quantify how often resistance evolves and then how much it transmits. And so this is a pretty old paper. It's 20 years old, but they found at least 20 different acquisitions of the SEC-MEC element. So the SEC-MEC element is a genetic element that makes a Staph aureus into a methicillin-resistant Staph aureus. And so this was one of those early papers that showed that MRSA is not just one thing, but it's actually evolved many different times. There's many different independently evolved MRSA strains. Okay, so both evolution within a host and transmission are important, and we can imagine that happening sort of in a transmission chain or in a phylogenetic tree, and these things are related. So here, you know, we can imagine that, say, this person is infected with a susceptible strain of, you know, Staphylococcus or E. coli or HIV, any of those things. They pass on their infection to this person. They're infected with a susceptible strain. They pass it on to this person that is initially infected with a susceptible strain, but then maybe this person is treated, and maybe inside this person, we get evolution. And now the strain has become resistant to a certain drug. Okay, so the red here indicates that it's resistant. The resistant strain is passed on to this person and to this person and this person. And then maybe here, this person is maybe infected initially with the resistant strain, but then it reverts to become susceptible again. That sometimes happens too. Sorry, that writing is not very clear here. Okay, so in the susceptible to resistant evolution happens in this person as well. And then that's passed on, and maybe here it's reverted again. So this is like a reversal to susceptible, and this one is resistant still. So when this is possibly what would have happened in reality, but usually we don't have data for all people that are infected with a certain pathogen, right? So we can imagine that some of those people, we end up getting a sample from them. Okay, we get like a blood sample or something else, or a urine sample if these are urinary tract infections. And so if we imagine that we have like 4 of those people, we have a sample from the pathogen for a study, and we sequence these samples. Then from these sequences, we can build a phylogenetic tree. And it may look something like this, where these 2 people, this one and this one, their pathogen strains are closely related to each other because they stem from a common ancestor in this person. And also they share the resistance. And that's because that resistance strain was passed on to different people. But then this one, the blue one that we sampled, also has resistance, but that resistance is due to an independent evolution event. Like this person is also resistant, but due to a separate evolutionary event. Okay, so that is just to, what I'm trying to explain here is that evolution and transmission are both important, and we can see traces of that when we look at phylogenetic trees of pathogens. If we see resistance, if resistance evolves multiple times, we can see the resistance appear separately on tips of the phylogenetic tree. Okay, now one thing that I haven't said anything about yet is what is the genetic mechanism of drug resistance evolution? And in bacteria, there's different ways that this can happen, well, depending on the bacterium. But generally speaking, for things like E. coli and Staphylococcus, we can have a transfer of genetic material in terms of, for example, a whole plasmid can be transferred from one bacterium to another. Or it could just be a single gene that is transferred from one bacterium to another. Or there's no transfer, but the genome mutates, at a replication event. Okay, so we can have, this is horizontal gene transfer, and this is mutation. Okay, and in something like HIV or tuberculosis, this is the only thing that happens, basically. But in something like E. coli, a lot of drug resistance is due to horizontal gene transfer. However, what's important is that horizontal gene transfer or mutation can both lead to this kind of situation where, say, this person got infected with a susceptible strain, and then due to horizontal gene transfer, it acquired a plasmid or a gene, and it made it resistant. And now that strain with the resistance gene or the plasmid is passed on to this person. Okay, so this one also has the resistance strain. And in this case, where like the donor has the resistance and this one doesn't, maybe that's because the plasmid or the gene was lost again. Okay, so even though, you know, in terms of the genetics, we may have different ways that the bacterium can become resistant, on the phylogenetic tree, that may look the same. Okay, so one big question for people who are working in public health and people who are worried about drug resistance, antibiotic resistance, all kinds of resistance is, how big is this problem actually? What fraction of the E. coli, the HIV, the TB, whatever pathogen you're interested in, is resistant to which drug? And what determines that number? So obviously, if say 1% of all E. coli is resistant to a drug, that is a smaller problem than if 20% is resistant to a certain drug, right? So we would like to know how much resistance there is, and would like to know what determines that number. And if that number, you know, is higher than we want it to be, which is usually the case, what can we do to make it go down? So for example, one of the something that I've worked on recently is ciprofloxacin resistance in E. coli in the UK. And in that case, about 15% of all E. coli infections in the UK are resistant to ciprofloxacin. Okay, so 15% is probably higher than we want it to be, but so we can measure that. People are measuring that, and a big question is, why is it 15%? Why is it not 20% or 100%? And what can we do to make it go down to 10% or 5 or 0? And one sort of, if we make evolutionary models and you have something that's at 15%, right? And this is time. Now, in most models in evolution, you know, we have this thing that we call, you know, survival of the fittest, right? Now, there's a lot of problems with that phrase, but I think it's what a lot of people know about evolution, is that whatever is more fit will take over the population, right? So if the resistant strains in UK are infecting more people, then over time, you would expect that 15% to go up and reach 100%. But if the susceptible strains are fitter and they are infecting more people, then you would expect that 50% to go down towards 0. But in reality, what we see is that it's pretty stable at 15%. And so there's currently no good explanation for why it's stable at 15%. Is there something magic about 15%? We don't know that. So that's an interesting, I think that's an interesting question, because if we understand why it's at 15%, hopefully we can also understand how we can make it go down. Okay, so we currently don't have a good explanation or a good model for how much resistance there is. And this means that we can predict how much and how fast an intervention will change a fraction resistant, right? So say in the UK, we would want to change that 15%, we want to make it go down. What do we need to do? Do we need to stop using ciprofloxacin altogether? Do we need to reduce how much ciprofloxacin we're using? Should we, and then if we do that, do we need to wait a month, a year, 10 years before that number will change? These are things that we currently don't know. And that's a big problem for public health, because, you know, if you work in public health, you want to be able to say, like, do this intervention and you know, it will hopefully have this result. Okay, so I want to answer those questions, and I want to make a model that can explain the what we currently see, the observations that we can make. So one important observation is this stability of resistant and susceptible strains. So the UK is here in dark pink. It's this line here. Am I saying that right? Yes, this one. So it's at about 15%. You know, early in the 2000s, it was lower, it went up a little bit, and then it became stable. All these other lines are different countries in the European Union. And what you can see is that they're not all at the same level, but they're all pretty stable. Okay, and so that is an observation that is not just true for ciprofloxacin in the UK, but it's true for many different countries, and it's actually true for other pathogens too. So if we make a model that can explain the level of drug resistance, it has to be able to explain that stable level. Okay. The other thing that has been observed by many researchers is that how much resistance there is in a certain country depends on how much of the drug is being used. So here, these are again countries in the European Union, including the UK, even though they're no longer in the European Union. Here on the x-axis is how much quinolones are used. Ciprofloxacin is a drug of the class quinolones. And so, you know, countries like Italy, they use a lot of quinolones, and also they have a lot of resistance to quinolones. This is E. coli. Okay. Countries like the UK and the Netherlands and Denmark, they use less quinolones, and they also have less resistance to quinolones in their country. Okay, so this is a second observation that any model that tries to describe the levels of drug resistance has to be able to explain. We do more treatment, we get more resistance. And then finally, this is a third observation, and this really comes from work that has been done by students in my lab. And I apologize, I should have had their names here. This figure comes from a paper that is on the bioarchive by Florentine van Nauhaus, Mary Gracie Anthony, and Faye Orcalis on bioarchive 2022. So you can look it up. And so this observation, again, this is E. coli, and this is resistance to ciprofloxacin, and this is a phylogenetic tree of part of some E. coli strains from the United Kingdom. And so what you see here is that resistance occurs on different parts of this tree, here and here and here and here, here and here. And so that suggests that it has evolved multiple times in the way that I showed in the drawings a few slides ago. Okay, so we have three observations. We have a stable level, we have a level that depends on how much drugs are being used, and then we have multiple independent evolutionary origins of the resistance. This is just to show that this is not just true for E. coli. So here, this is data for HIV, and here the different colors are resistance to different drugs, and you know, these lines are pretty flat. The green one is actually not entirely flat, but again, like the level of resistance is pretty stable. And then this is a different way of showing here each of these little rows is a different origin. It's a different cluster on the phylogenetic tree of drug resistance. And so in this paper, I think they identified something like 80 different origins of drug resistance on their phylogenetic tree. So again, for HIV, just like for E. coli, we see a stable equilibrium, but many different origins of resistance. Okay, so after staring at this data for a long time, I had an idea. The idea was, can a model of mutation selection balance explain the data? And mutation selection balance is a classic population genetic model. I've used it for other types of problems in data. And until very recently, maybe like a month or two ago, I didn't consider using it for this situation. And the reason I didn't consider using it is because mutation selection balance, as the name suggests, only has two ingredients, mutation and selection. And in this case, selection means negative selection. So mutation selection balance is used to explain the existence of deleterious mutations. So for example, in human population, every time a new kid is born, that kid will have new mutations compared to the parents. And some of these mutations will be deleterious. They're bad for us. Okay. And so mutations are created every time our DNA is copied. And then there's negative selection on these mutations. So the mutations will be what we call purged out of the population by selection. So in this model, new mutations are created every generation, and then there's selection against these mutations that will keep them at a low frequency. In drug resistance, we know that there's not just mutation and selection, but there's not just mutation and selection against drug resistance. We know that there's also selection for drug resistance at the within host level when people are being treated with drugs. And so the mutation selection balance model, seemed to be too simple. But I think I found a way to make it work. And I think it can explain the data. Okay, so in this new model that is based on this old model, I think there's a balance between new resistant strains that evolve, that are being created. So this is within host evolution. So that continuously creates new strains, and this is due to positive selection for resistance inside someone's body if they're being treated. And so that's what we see in HIV very often, but we know that it happens for all these pathogens. But then that's balanced by competition with susceptible strains. And so this basically happens in the population at large. So if we have a lot of people, most of them, most of the time, are not being treated with whatever drug we're interested in. And so at the population level at large, the susceptible strains may always be more fit than the resistant strains, because most of the time there's no drug. And so the drug resistance, if there's no drugs, we usually assume that the susceptible strain will have the benefit, will be more likely to replicate or more likely to transmit. And so what we can model is a situation where these new strains, occur all the time, and they increase the fraction of resistance in the population, and then the competition with the susceptible strains will decrease the fraction resistant, and the result is that we see an equilibrium. And if the competition with the susceptible strains is such, if that force is very strong, maybe because the resistance comes with a very strong cost, then, if these arrows are sort of stronger, this will go lower. But if we have a lot of treatment, so that there's a lot of resistance evolution happening in treated hosts, that may make these up arrows stronger, and, the equilibrium will be higher. So let me try to go over the math. And I know that there's a lot of people out there who, who worry that mathematical models are always very complicated. And I think in this case, the math is not that complicated. So I'm going to try and explain it here. Okay, so first I have to sort of create a population. So bear with me, I gotta do some drawing. Okay, so a whole bunch of people. Okay, they're not very pretty. Oh, sorry, their heads are like falling off. Okay. So we're interested in the fraction. Well, first of all, for this model, I'm only interested in the people who are infected with the pathogen of interest. So all of those are infected. Okay. And now of those people that are infected with the pathogen of interest, I'm interested in what fraction of them is infected with a resistant strain versus the others are infected with a susceptible strain. Okay, so let's assume for now that some fraction is infected with a resistant strain, and we'll study how that number will change over time. So let's assume for now that these are infected with the resistant strain, and all the others are infected with the susceptible strain. Okay. And so that fraction resistant, we call it F. And the fraction not resistant is is 1 minus F. Okay, and we're interested in how this number changes over time. The fraction, so fraction of the pathogen population that is resistant. Sorry. Okay. So there's these two forces, right? New resistance comes up, evolves, and then competition makes the resistance go down. Okay, so first, new resistance can evolve only in this part of the population that is currently not resistant. Okay, so new resistance can evolve in the part of the new resistance can evolve in a part of the population that is currently infected with a susceptible strain or non-resistant strain. So that's the fractions 1 minus F. And then with a certain probability, they are being treated. So let's say, those are the ones being treated. And then out of those that are being treated with the drug of interest, a fraction of them, the pathogen will evolve to become resistant. And that fraction is E. Okay, so maybe this one, maybe there's 4 of them being treated and one of them evolves, the pathogen evolves resistance. So maybe E is like a quarter. Okay, so new resistance evolves at this rate, 1 minus F times T, the probability of being treated, times E, the probability of evolving resistance given treatment. Okay. At the same time, the ones that are resistant, so these ones, they are suffering from competition with the susceptibles. And how I imagine that happening is that there's a certain probability that these resistant strains sort of fail to reach the next generation, fail to infect new hosts, because the susceptible ones are better at infecting new hosts. And so we lose resistance, or like reduction of resistance by a factor F, so the ones that are resistant, times C. And C here is the cost of resistance. It's how much, it's sort of a measure of that competition from the susceptibles. If the cost is strong, the competition is strong from the susceptibles, and we lose resistant strains. Okay, so the change in the fraction resistant can be written as F prime for change can be written as 1 minus F times T times E minus F times C. So again, this is new resistant strains evolving, and this is resistant strains basically losing out in the competition for new hosts. Okay, so now the next step is to say, well, if F prime, that's the change in the fraction of resistance. If F prime is 0, we're at an equilibrium. If F prime is 0, the newly resistant strains balance out exactly the loss of resistance due to competition. And so if we set F prime to 0, that must mean that this must be exactly as big as this. So we get 1 minus F times TE equals F times C. Okay, so here we can do some simplifications. The left part here, we can write it as TE minus F times TE. That must be equal to F times C. And then all the things that have an F, we put it on one side, so we get TE equals FC plus FTE equals F times C plus TE. And then we divide by this on both sides, so we get F equals TE divided by C plus TE. Okay, so there's an equilibrium in this model where the frequency or the fraction resistant is equal to TE divided by C plus TE. And TE is the probability of treatment times the probability of evolution of resistance or acquisition of resistance given treatment. And C is the cost of resistance, how strong the competition is from the susceptibles. And so that means that if there's more treatment or more evolution, if T or E or the combination of the two is high, then that increases F. But if the cost is high, that decreases F. And so in this model, what you get is that if TE goes up, you get more resistance. So this is F, and this is T times E. If T times E is higher, you get more resistance. And then these different colors are for different values of C. So if C is very small, you get very high levels of resistance. If C is high, so if there's a strong cost, you get low levels of resistance. Okay, so I see that I'm running out of time, so I'll try to, so the model is simple, is mathematically simple, because we assume that resistance always comes at a cost. So the resistance strains always pay that price C, which makes that the fraction of, if there was no new resistance evolving, resistance would always go down. Okay, this model basically makes a bunch of predictions, and I think these three are interesting, and I want to share them, and then I'll stop. Okay, so the first model prediction is that even if the fraction of resistance is stable, it's stable because there's new resistance evolving all the time. And so I actually looked at a lot of data. This is data for ciprofloxacin resistance in the UK. And every color here is a different sequence type. Sorry, the gray here, this is the susceptibles, and all the colored ones is the resistant ones. And the different colors are resistance that is carried by different sequence types. And so what you see, for example, here is that this blue sequence type, it seems to be very important. And then it sort of disappears. So that suggests that resistance strains appear and then disappear again. So that fits with the model. Overall, resistance is fairly stable, but who is carrying the resistance is changing over time. Okay, the second prediction is that the cost parameter, C, will determine how long resistance strains can survive. If the cost is very strong, resistance strains, will appear and disappear very quickly. And that means that if we do interventions, like if we change how much treatments, how much we are treating people, how fast an intervention will have an effect will depend on that cost. And so, for example, here is data from an intervention study where in a hospital, they decided to reduce how much ciprofloxacin they were using. And then they see also a reduction in how much ciprofloxacin resistance there is. And actually, this is sort of surprisingly fast. In just a couple of months, they see the effect. And so that suggests to me that ciprofloxacin resistance must come with a fairly high cost, because the resistance strains that were responsible for this high level of resistance must be disappearing fairly quickly. Okay, and then a third prediction, which I haven't been able to test, is that if you compare between high and low resistance countries, so again, for example, Italy versus the UK, the difference should really be in the number of new resistance strains evolving. If my model is correct, then countries with a lot of resistance should have a lot of new resistance strains coming up all the time because so many people are being treated. Okay, I don't really have time to show you all of this, but I think this all works for horizontal gene transfer basically in the same way as it works for mutation, which is exciting. So in conclusion, I proposed here a simple mathematical model that can explain three key observations. It can explain the stability of the level of resistance. It can explain the strong correlation between treatment and fraction resistant. And then it can explain why resistance seems to appear so many times independently on the phylogenetic tree. So with that, I'm going to stop. Thank you for listening. I need to thank my lab, my amazing students who are willing to listen to me talk about this all the time, and many amazing colleagues at SF State and beyond. The evolutionary biology community is really lovely. And thank you, Scott, for hosting.
That was fabulous. Thank you. It was really great. And I've just got to give you props for just going through the math so clearly like that. That's just so helpful when people do that for the students, but it was perfect. So we have a question from Lauren.
Hi there. That was really interesting talk. Thank you.
Thank you.
So it sounds like is this like ready to be used for public health initiatives? For example, like could hospitals establish like an enhanced genomic surveilling for drug resistance where they could then create like a collaborative network and adjust their antibiotic administrations based on, those evolving resistance patterns?
Yeah, I would love to to sort of get genomic data from a situation where an intervention is being done. I'm not aware of a situation where that has been done. Like, so there's intervention studies and there's genomic studies, but I haven't seen them combined. So I would be very excited to see that. Is this ready for guiding public health interventions? On the one hand, I want to say yes, because I'm convinced of my own work. But on the other hand, I would never trust a single scientist. So it would probably have to be replicated
Yeah.
by others before anyone really trusts it.
Yeah. All right. Thank you so much.
Thank you.
More questions? I have one, but I'm going to throw it out there. Riley.
Hi. Yeah, thank you for the great talk. I had a question. So ultimately, E. coli is very easy to culture in lab. Like I had missed the first part of the talk, but like I don't necessarily know your background. But ultimately, though, like have you done in vitro validation of these findings? Because like given that E. coli is very easy to culture in the lab, I mean many labs do it routinely. Like have you or a collaborator looked at these mechanisms of resistance? Because it seems to me, like given what I know about evolutionary biology, that ultimately, like the resistant strain should always win, but with the data you've shown, that's not the case. So ultimately, like what is the molecular mechanism for this kind of like steady state competition between susceptible and resistant strains? Because I didn't see any validation of that whatsoever.
Yeah, thank you for that question. So actually, like a lot of the drug resistance mechanisms have been validated by others already, and so I can basically just build on that previous work. So there's so a lot of drug resistance in E. coli is very well studied, and so for example for ciprofloxacin resistance, it's mostly due to very specific mutations in the gyrase A gene that have been well studied. And then other than that, people have studied the costs of resistance. And so in many cases, for many pathogens, sort of pathogen drug combinations, it's been shown that there's a cost when the drug is not there. Not for all. Like in some cases, people have not been able to find a cost. And that could be either because there is no cost, or it could be because, the cost is so small that it's hard to measure, or because it's hard to measure. Maybe there's a cost that we cannot measure in the lab because it's specific to being in a human host. But there's a wealth of literature on these things. And I do not culture, I do not have a lab in that sense. I only do the computational work, but in this case, I think that's really okay because so many other people have done a lot of this work. So I don't need to repeat that.
Okay, thank you.
Thank you.
Yeah, so I had a question about this. And I've been watching the COVID disaster unfold, and knowing what I knew about HIV, I was like, this isn't going to last very long, these vaccines, for similar reasons that the drug resistance wouldn't last. I mean, the evolution is different, but you have all these other issues. But then you have these other completely amazing things going on. I see the same graphs about well, how many strains, how many are evolving resistance and new ones coming in, you know, all that kind of stuff. And what I see is, in addition, you have in-patient evolution like that. You have the addition of the vaccines, which are not drugs, but, you know, might have similar evolutionary pressures. And you have this amazing antibiotics that have been given, where you, or not antibiotics, they're antibodies, like the monoclonals, where you actually are giving them to patients. And there's two kinds. There's one that, you know, like just doesn't do it, it just binds to it and then causes it. But there's other ones, I think it's molnupiravir or something, that actually increases the mutation rate within the patients, which sounds like an amazing way, a beautiful way, and totally dangerous way to increase one of your variables, which is the number of increasing resistance strains out there. Have you thought of, I mean, there's such a wealth of genomic data for the COVID. Have you thought of taking your model to any of these systems? Because it seems like if you want genomic data, there you go.
Yeah. Well, so no, I haven't taken it to COVID, in part because there's so many people working on COVID. I sort of like to work in a space where I'm not directly competing with other people who have, like, you know, better funded labs and everything. And, you know, also like there's, like, honestly, like I've been so impressed with like the work that's being done on COVID. But okay, so here's the thing. So in terms of like vaccines, what is really different between COVID and, like, say that what I'm working on is the vaccine spread is so high. You know, like in my model, I can assume that most of us, most of the time, we're not being treated. Like most of us are not currently on ciprofloxacin, right? And even if we're like some infected with E. coli, and most of us carry E. coli all the time, like most of the time we're not on that kind of treatment. Whereas with the vaccines, you know, most of us are vaccinated. And so that makes for a very different situation. And then in terms of like resistance to some of those, to either the antivirals or the antibodies, or like, yeah, I think that could actually work the same way, and it'd be interesting to look at that. That's probably a good idea, because there's a lot of data. But, you know, in terms of like E. coli, you know, one of those last plots that I showed that I didn't really have time to talk about a lot, the one that had like the many different colors, that's like based on 37,000 E. coli genomes just from the UK. Like I wasn't able to do all of Europe because it was like so big, so I limited myself to the UK. But like there's thousands and thousands of E. coli genomes out there, so there's definitely interesting genomics that can be done.
Oh yeah, no, that's true. I have a vested interest because I have to take the monoclonals because I can't be vaccinated because of the medication I'm on. I mean, I can be vaccinated, but it just doesn't work.
Yeah.
Because of the kidney transplant, blah, blah. So I've seen this happen where I took a monoclonal for a while, then they said it's no good anymore. And then they have this other one that's driving mutations, which may actually be driving evolution. So it's personal. One other thing, I had a question about your graphs. I see this a lot with the proportional graphs. But what happens is when you see the proportion of something disappearing, right? The graph only goes to 1. So like it's from 0 to 1, right? And so it could be that it's actually getting smaller, or it could be that other things are increasing and it's staying exactly the same. And I keep getting frustrated with this because it's all dependent on how many infections there are currently, your interpretation of that. And so I know it's probably, maybe yours is very steady state, and so it's always the same number of infections. So that would really be true. But in COVID, it's not true.
Yeah.
Because then you have waves, and then you're like, well, it didn't actually leave. It's just that another one just went bananas, and there's no direct competition between the two. And that really is interesting because like they're not really competing because one's just going crazy, but this one's still bumping along somewhere. So do you ever see anyone like bring in the absolute numbers into their graphs, or does this matter at all? Or, you know, I just
that's a good question. And I think you're right. Like in COVID and in some other cases, there's really not, you know, it would really be sort of unfair to just plot frequencies instead of like the whole population. I think in E. coli and in some of these other pathogens, it's really quite stable. Like I don't think the number of E. coli infections is going much up or down. So I think that's okay. And I actually also think that like, I'm also sort of pragmatic in the sense that like that's a system that I can more easily work with. But I think you're right. It's a valid point. Like, is there actually evidence for competition or is it something else? And in this case, I think there is. If there wasn't, the number of E. coli infections would just, and I think, I mean, sort of competition, it's sort of, I mean, I actually think that competition, like it's sort of a nice word to use because it's like something that we can imagine, but it's sometimes a little, like, it's not like they're directly fighting
like
each other.
Yeah.
Yeah.
Like boxing.
Yeah. But, if you have an infectious disease that is present at a stable level, right? That means that the level that it's stable at will be a level where every infection on average replaces itself. So the R value becomes 1. That's what it means to have a stable level, right? You're infected, on average, your infection will lead to 1 new infection at the next time point. And so that must mean that if I was the only person that was infected with E. coli, then my E. coli would infect a bunch of other people, right? Because the R naught is much higher than 1. Right, like the infection, like if the population was not infected at all, like the pathogen infections would go up. The fact that they're not going up means that they feel each other's presence. Yeah, I didn't do a very good job explaining that.
No, it's a good question because it is like, it's not necessarily like the way we think of competition is maybe like, well, if you get it, you can't get the other one. And in that way, it's out-competed. But what it means is it transmitted before the other one could get there, which means they're not directly fighting. It's just that you can't get both at the same time. Usually don't. You get one thing and it takes off.
Yeah.
That's
And of course, some of these pathogens, you can have multiple infections, but there's a limit to that. Yeah. And then some people, no, absolutely. Yeah.
Yeah, it's good to think of it biologically in addition to like, math and stuff, because you're like, wait, how does this actually happen?
Yeah, that's true. Good point.
Do we have any other? I could ask a lot more questions. I love this topic, but here, Marisol. Yeah.
Amazing talk, Dr. Pennings. I just had a question regarding your constant variable and understanding how it's normalized when it could be increasing or decreasing. That'd be super helpful for me to understand. Thank you.
Wait, Marisol, can you say that again real quick? So you're interested in like why? Wait, can you just ask the question again? Hopefully I'll understand it better.
So within your mathematical model, you have cost as a C variable.
Yeah.
And I know fitness cost, I'm assuming, could go up or down. And in this case, it's normalized to a single number or variable in this case. So I'm wondering, was this based off of literature that I don't know yet?
Yeah.
Explain that. Thank you.
Yeah. So I assume that there's a constant cost, like a fixed cost. So for example, we could imagine that, sort of regular E. coli is 5% better at transmitting compared to ciprofloxacin-resistant E. coli. Or rather, we usually measure it the other way around. Ciprofloxacin-resistant E. coli is 5% worse at transmitting compared to ciprofloxacin susceptible E. coli. And so I assume that for a given drug, I assume that there's a fixed difference in how transmissible these two types of strains are. In reality, that's probably not entirely true. You know, if resistance evolves multiple times, each of them may have like their own slightly different cost depending on the genetic background. And also the cost may be different depending on like what type of host they're in. But I think to understand the big picture, it's okay to work with a fixed average cost of resistance. So I sort of assume that that's fixed. Not sort of, I assume it's fixed.
Thank you.
Thank
Sometimes
you.
the models have to be simplified so that they can work, but that's pretty cool. Okay, well, it's 1:10, so I think it's probably time to wrap it up. But I love that talk, and it's just such a fascinating subject to think about, and hopefully those models can really help people control infections and think about the evolutionary aspect to it, which is often ignored in public health, and definitely I thought was ignored very immediately in COVID. So I had all sorts of questions about where other reservoirs and hospitals and stuff like that, but that's for another time.
Another time.
Another time. All right. Well, thanks a lot, and I hope we'll get a chance to talk in person sometime. And thanks everyone for coming to this great talks. I'm learning so much. I'll put this video online.
Thank you. Thank you. Bye, everyone.
Bye.